Well, thank you. Thank you for those kind words, and it is very nice to be here. I've been to CPRI several times. Once I remember the first time was when I was a resident, so way back in the late 70s, and then have made several visits over the years. So it's great to be here and to meet you and have a chance to talk to you. The title, I know, is a little macabre, but I'll just do this slide first, which is my financial disclosures. These are funding sources that support the grants that I'm involved with. I get royalties from Guildford Press. For a book I wrote, actually I used to get royalties. The book stopped selling. I saw a mom a couple of weeks ago who came in and got a copy from Value Village, so she said, I got it from Value Village. It was like $1.50. Oh, geez, no wonder. So what I'm going to talk about is this concept of the Valley of Death, and what do we mean when we refer to that term in the context of biomedical research. I'm going to give 3 examples in the autism field about traversing the Valley of Death. One that I think we've done relatively successfully, one where I'm worried that we're prematurely traversed the Valley of Death, and the third where there's a lot of excitement and anticipation about this particular traversal. And going to use this really as an example to talk about translational medicine in the autism field in particular. So this is where the Valley of Death comes from. It's from a line from the Charge of the Light Brigade by Alfred Lord Tennyson. Into the Valley of Death rode the 600 during the Crimea War. It's actually 673, but that wouldn't work in the poem, right? Into the Valley of Death rode the 673, so he took a poetic liberty with it. The Valley of Death can also refer, of course, to the Valley of Death in California, which is another. And the person who coined the term actually didn't really define which Valley of Death he was talking to, the one in California. But since it was an American, I bet you it was the California Valley of Death. But since I'm Canadian, it's going to be Tennyson's poem. This Butler first used the term, and it's really caught on in the medical field in general. First used the term in 2008. Really refers to the failure of basic science findings to have an impact in clinical practice. And there are lots and lots of different reasons for this particular failure. There's a dreadful sort of lack of communication between academia and industry. There's an extraordinarily tortuous process by which clinical trials need to be approved, and then findings from clinical trials actually then make it to market. There's a real paucity of translational scientists. So we have basic scientists and we have clinical scientists, but we don't have a lot of translational scientists, that is, those who sort of have a feet in both camps and are able to use findings from one to the other to make a difference in clinical practice. There's one very striking series of events in recent years is the extent to which the pharmaceutical industry has actually withdrawn from research and development altogether and is leaving more and more research to universities, and then taking findings from universities and then marketing it from that point of view. And even though the pharmaceutical industries have really stopped providing a lot of money to universities, much less than they used to. And then there's the whole issue of intellectual property, which is a real nightmare, particularly in the university setting. This is a very famous paper that was written by Green, and let's see, that's also 2008, talking about this line where it really takes about 17 years to turn 14% of original research to the benefit of patient care. So what he's saying is that only 14% of all the research that we do has clinical impact, and it takes 17 years from the point at which that particular finding is published to having an impact in clinical science. And this particular paper is widely quoted as really an example of the problem of the sort of valley of death in biomedical research. And what he, this just really illustrates, you know, these are all the studies that are originated in research, and then there are findings that come along that are submitted to articles, and then they're accepted, and then they're finally published, and then they get cataloged in a synthesis or a meta-analysis, et cetera, et cetera. So there's this falling off. This is the valley of death where you lose findings as you go along from the original research to its clinical implication. And I think this is a real problem in the ASD field because we have two valleys of death, as it were. We have one valley of death, which is the basic science finding to a clinical application, but then there's the clinical application that we determine to be useful through randomized trials, for example, to then making an impact in the community. So the effectiveness of a particular intervention of some kind. So we have these two. And I think another reason is the pace of technological advances in autism, particularly in the genetics field, has been absolutely breathtaking. I mean, now we can sequence somebody's genome for maybe $5,000. It won't be long before it's under $1,000. It used to cost millions of dollars maybe 5-10 years ago. So the pace of that technological change is absolutely enormous, and it leaves, frankly, a lot of clinical scientists like myself behind, so they're not really being able to understand. And then there is this paucity of clinician scientists in recent years. We're a Canadian academia, and in fact, academia in North America altogether isn't producing as many clinician scientists as we used to a number of years ago. I'm going to talk about 3 examples of traversing the valley of death in ASD. One, some comments about DSM-5, whose time has come, as it were. I'm going to talk about clinical microarrays for copy number variants in ASD as a first-line test in ASD, and then I'm going to end up with a brief discussion about some new drugs for ASD that are targeted to the genetic loci that have been identified by these copy number variants. So DSM-5, and this part of the talk I've sort of entitled 2 steps forward and 1 step back. So really quickly, I just want to review very briefly DSM-4, the thinking behind the changes in DSM-5, talk quickly about the evidence for those changes, and I want to give this really as an example of how the field has been able to translate a very large body of work, which started almost 1994 with the publication of DSM-4. So we're not quite 20 years post that, but we're getting to the 17-year period. So this is a good example of that 17-year period where almost immediately after DSM-4 was published, people started to evaluate its usefulness, and we're just now getting to change that into DSM-5. So you all know DSM-4. I'm not going to spend a lot of time with this, but we've got 3 symptom domains: impairments in social reciprocity, impairments in verbal and nonverbal communication, and a pattern of repetitive stereotype behaviors. We've got 4 items in each of the 3 domains, 12 in total. To be above the cutoff, you have to have 2 in the social, 1 in the communication, 1 in the repetitive domain. You have to have 6 in total, and your age of onset has to be under 3 years of age. And then we've got all these DSM-4 subtypes. We've got autistic disorder, Asperger disorder, PDD-NOS, Rett's disorder, childhood disintegrative disorder. Now, it didn't take long before people began to have concerns about DSM-4 post-1994. One was that a feeling that it was a bit over-inclusive, particularly with this concept of pervasive developmental disorder not otherwise specified, which is really a sub-threshold form of PDD. But a lot of individuals were getting the diagnosis of PDD-NOS, and a lot of concerns about whether it was being overdiagnosed and it was over-inclusive. clinicians really, there's some studies showing that clinicians weren't able to apply the criteria for PDD-NOS and Asperger's syndrome reliably so that everybody could agree, you know what, this kid has autism, this kid has Asperger, this kid has PDD-NOS. If you looked at the extent to which clinicians agreed on the assignment of the subtype, there was a lot of disagreement, and in some cases it was almost random. And even more damning was that these subtypes didn't appear to differ with respect to etiological variables like causation or neuroimaging or outcome or response to treatment. So even though you might spend a lot of time deciding what type of PDD a kid had, you know what, it didn't really seem to make a lot of difference clinically. And then there's this, I just want to emphasize that the DSM-IV understanding of ASD is the famous autism triad of social reciprocity, communication, and repetitive behaviors. Well, the first thing that sort of became apparent was that actually it wasn't really a triad. So there were a number of factor analytic studies that were done showing some studies showing, for example, that the ASD phenotype was unidimensional. It was one phenotype. It wasn't a triad. It was what's a one thing? What's a uniad or something? Unitary something. I don't know. Another group of studies showing that actually was made up of multiple different phenotypes all coalescing together. But really, probably the most consistent finding from the factor analytic studies is that the social communication domains always occurred together. That you couldn't really have a social domain that was independent of a communication domain. You can have a social communication domain independent of language, grammar and vocabulary, but you couldn't really have social interaction that was independent of communication. So instead of a triad, we really got a dyad, was a really important conclusion from a lot of those factor analytic studies. Okay, so then the APA establishes a DSM-5 committee. They move from they move from Roman to Arabic. That's the first important change. There's a subgroup of the Neurodevelopmental Disorder Working Group that works on autism ASD. You know, these are international experts in diagnosis and measurement and clinical expression from around the world, and they specifically used people who weren't in DSM-IV. So I happen to be lucky enough to work in DSM-IV, and so that's what I tell people. That's why I wasn't invited to be on the DSM-5 committee. They did a thorough review of the literature. They published reports for publication that are on the DSM-5 website. They conducted field trials, and they've proposed some new criteria for ASD. So I'm taking this from the DSM-5 website, January 2011. So instead of the different autism spectrum disorder subtypes, instead of autism, Asperger, PDD-NOS, there's going to be one category called ASD. And the argument is that the argument for doing this, to collapse them all together, is that all these different subtypes don't differ from each other on etiology. There's also an important need to reduce the overdiagnosis, so that too many kids were getting the PDD-NOS diagnosis. We can reduce the overdiagnosis if we just go with something called ASD. A concern to make the diagnostic criteria more developmentally appropriate. So, because in DSM-IV there was a concern that the criteria were more applicable to younger kids and much more difficult to apply to adolescents and adults. So they tried to simplify it. And then there's this notion of, you know, in DSM-IV it had to be under 3 years of age. Sometimes it's hard to, hard to remember if it was under 36 months. Was it 35 months or 38 months? You know, that's not such an important threshold. Don't make it so rigid, they would argue. Let's just say it has to be present from early childhood. The radical thing is that instead of the triad, we're going to have two dimensions called social communication and repetitive stereotyped behavior, based on all these factor analytic studies that were published. And there's going to be a spectrum. Again, another important finding that everybody was coming up with in the phenotype studies is that it is a spectrum disorder ranging from mild to severe, but we've got 2 dimensions to sort of to characterize on a dimensional manner social communication together and repetitive stereotype behaviors. And then they're going to add these clinical specifiers that haven't really been defined or discussed very well. So you could have, nonverbal autism spectrum disorder or higher functioning autism spectrum disorder or something like that. I don't think that's clear at this point. And these are the criteria for the social communication domain. Somebody has to meet all 3 of the criteria. Here are the items for the repetitive sensory motor behavior domain. You have to meet 2 out of the 4 criteria. And this is the kind of idea, you can't see that very well, I'm sorry, but this is the social communication domain. This is the repetitive stereotype behavior domain. You could have somebody who with severe impairment in social communication and much milder impairment in repetitive stereotype behavior. So this is how they want to sort of begin to characterize individuals by incorporating this dimensional component that was so evident from a lot of these phenotype studies. Well, people have been trying to validate and evaluate the DSM-5 criteria in the last year or so, and you know what? It seems actually to be working reasonably well. Studies confirming that we've got 2 dimensions instead of 3. There was one paper that raised a lot of fuss about possibly missing some cases with Asperger's syndrome and high-functioning autism. The Fraser study showed that as well. But I want to emphasize, and this hasn't been appreciated really, that these papers where there's this concern about missing cases was published before, and the work was done before the DSM-5 criteria were modified in January of 2011. So those papers aren't really a fair evaluation of the DSM-5 criteria. In the last year, a couple of really important papers indicating that the DSM-5 criteria are actually, in fact, more accurate than DSM-4, that the kids who got the DSM-4 criteria for autism will also meet criteria in DSM-5. So this has been a real concern for people. Clinically, a lot of parents are concerned, do I have to get my kid re-diagnosed according to DSM-5? This paper suggests no, that there'll be that continuity. And also concerned that the research that we did using DSM-4 is now going to be irrelevant because we've changed the criteria. And this paper suggests, well, actually, the research we did with DSM-4 is still relevant when using DSM-5. I was asked to do an editorial, and it seemed to me, sort of evaluating all this, is that, you know what? This is a simpler model. This 2-dimensional model does make sense. We're not going to need as many experts to make the diagnosis as we used to in the past. So that's the 2 steps forward. It's better appreciation of the evidence, and it's going to be simpler. So that's my point about there's less need for expert clinicians. Maybe this will get us rapid access to clinical services. A concern, certainly in my community, is that some kids with Asperger's syndrome and PDD-NOS are denied services because they don't have, quote, autism. So now there's not going to be autism. There isn't going to be Asperger, there isn't going to be PDD-NOS. Everybody's going to get the one diagnostic category, and you can't discriminate against people getting services because they have one of these particular subtypes. Hopefully, there'll be shorter diagnostic odysseys because it's still a problem. Parents get concerned about a kid's development still at around 18 months of age. Average age of diagnosis in Canada is around 3.5 or 4 years of age. That's much too long a time period, and maybe this process with DSM-5 is going to simplify that and shorten the diagnostic odyssey. I hate the term PDD-NOS. I think it's a It's just way too open to confusion, so I will be glad to see this go the way of the dinosaur. But my concern about DSM-5 has to, and this is the one step backward question, is whether the one category captures the diversity and the heterogeneity that we see in the disorder. And, you know, it is, we all know how extraordinarily heterogeneous the condition is that you can have two people with the same diagnosis but looking very, very different. So this is just a slide from our study of kids with Asperger syndrome and high-functioning autism. And we studied the kids originally when they were between 4 and 6 years of age, and at the end of the study they were between 17 and 19 years of age. And this is using the Vineland socialization domain, looking at their raw scores over time. And this is the sort of the growth curve of the kids with Asperger syndrome, and this is the growth curve of the kids with autism. So at the beginning of the study, the kids with Asperger syndrome had better social skills than the kids with high-functioning autism, even though their IQ wasn't different. And they had, at the outcome at 17 to 18, they maintained their advantage. So they're different, right? Their kids with Asperger syndrome are doing better. But they're also the same in the context of having parallel lines, right? These are two parallel developmental trajectories over time. So when people say, well, is Asperger syndrome the same or different than autism? And the DSM-5 committee answered, it's the same. Well, it's both the same and it's different. You can't deny that there's a difference at the baseline, and you can't deny that there's a difference at outcome. Now, maybe autism and Asperger is not the best way to subdivide. There are other ways to look at the data hypothesis-free and to have a more empirically based classification. So this comes from a recent paper published in Pediatrics by Fountain and al. Actually, the senior author is Bierman. And this looks at social trajectories among kids with autism spectrum disorder from 3 years of age to 14 years of age. And they're using a statistical technique that groups together children that have similar developmental trajectories over time. And the basic point of this article is look at the different ways that kids with autism spectrum disorder, how different their developmental trajectories are over time. There's some kids that make very little progress in their social development over time. There are other kids that start off at the same spot, basically, with respect to their social functioning, then make rapid progress and then level off. There are other kids that are better at the beginning. They make rapid progress and they kind of level off too. There is enormous heterogeneity in the natural history of autism spectrum disorder over time if you allow the data to speak for itself. And I think one thing I worry about is the extent to which we're going to lose an appreciation of that heterogeneity by just having one category. So that's my example of a translational finding whose time has come. 2 steps forward, maybe 1 step back. Let's see what happens. What about translating their genetic discoveries into genetic testing? I'm going to talk about copy number variants as a source of genetic variation. So copy number variants refer to structural changes in our DNA. You know what? Our genes, our genes are full of holes. Little extra bits of base pairs are thrown in there. There are duplications. There are insertions. Like our DNA, frankly, looks like Swiss cheese, and little extra bits as well on the holes. I mean, it's not a smooth helix as you think of it in the textbooks. And these genomic variations, these structural variations, can be very rare or they can be very common. So we all have them, and some of them are very, very common, and they can arise de novo, spontaneously, or they can be inherited, transmitted from one's parents, and passed through different generations. And the argument is that when we started looking at copy number variants in ASD, we thought, well, okay, if we can find a rare copy number variant and it influences a gene that's expressed in brain and it disrupts the function of that particular gene, maybe that particular copy number variant could be a cause for autism in a particular child. So this is what sort of copy number variants look like. You have a reference genome, so a genome from a typical person. This is just a segment of a particular chromosome. And this is what a deletion looks like. In other words, there's a little section here that's actually deleted. It's not there. And the genes in there, instead of having 2 copies of every gene, which you would have typically, you have 1 copy of every gene. Here's a duplication. Here's the reference genome. You can see that this little section of that particular of the chromosome is duplicated in the individual with ASD, for example. So instead of having 2 copies of that gene, you have 3 copies of the gene, hence copy number variation. So the first paper that was published on copy number variants was in 2007. Genome-wide copy number variants was our paper that looked at copy number variants in a sample of about 1,000 affected SIB pairs with autism spectrum disorder. And in that, we found 1 copy number variant which crossed over or affected the neurexin gene. Since that 2007 paper, there have been hundreds of different genes that have been reported that one sees copy number variants in them in cases of autism more commonly than in typical controls. Neuroligand genes, SHANK1, 2, and 3 genes, I'm going to talk a bit more about them. Contactin-associated protein, PATCH-D1. It's like at least over 800 different genes have been shown to be affected in individuals with ASD by these rare, mostly de novo copy number variants. But each one of them is very rare. So each one of them only occur in, say, less than 1% of cases with autism. But because we've got 800 of them, they're accumulating week by week, and maybe we can explain 10% of cases with autism now who have these copy number variants in some of these important genes. Now, the interesting thing is that even though each one of them is very, very rare, they all have common, they're all part of a pathway. They all have common functions to the extent that they influence the way neurons grow together and communicate with each other. So, used to be, we think of the genes that were involved with autism were monoamine genes, serotonin, norepinephrine, dopamine, etc. No, we've thrown that out the window. The genes that seem to be involved in autism are those genes that influence the growth and development of neurons at a very early stage of development. And this is just an example of the 800 genes that we find. They're not randomly distributed in space. They're all related to each other so that the protein that one gene produces actually interacts with the protein that another gene produces that has also been found in autism. So even though it looks like a dog's breakfast, actually, if you look at the function of those particular genes, they're related to each other. So we've now taken this particular finding, and people are talking about using these copy number variants as a way to diagnose autism. So we can now, it looks like, we can have genetic testing to discover the etiology of the disorder. Maybe we can shorten the diagnostic odyssey. Maybe family planning. Say you've got one child in the family that has autism. Parents are wondering about whether they should have another child. They do this clinical microarray. They do the genetic testing. They find that there's a de novo, a spontaneous copy number variant. Well, if it's de novo, it's unlikely to occur in another affected child in the family. That could reassure parents that they don't, that they can go ahead and have another child without being worried about autism occurring in another child. So we've got this, we can now incorporate all these different copy number variants into something called clinical microarrays. And clinical microarrays have now become the standard for first-tier testing, for genetic testing in autism spectrum disorder. I'm just going to give you an example. This is the SHANK1 gene. So this is the presynaptic neuron, postsynaptic neuron. SHANK genes are these proteins here that sort of stick the two different presynaptic, postsynaptic neurons together so that they communicate with each other most effectively. Just going to give you an example of how powerful this genetic testing can be. Here's a family that have a mutation, a copy number variant in the SHANK1 gene. It starts with the grandfather who passes it on to his daughter. The daughter has the SHANK1 mutation. She's completely normal. She has a boy that she transmits that SHANK1 mutation to. He's affected with Asperger's syndrome. Another boy does not have the mutation. He's normal. Two girls, two daughters don't have it. Their kids are normal. This daughter does inherit the mutation. She has two boys, both of whom are affected. A daughter who doesn't inherit the mutation, she's normal. So this is an example of a mutation that acts like it's autosomal dominant with 100% penetrance in males and 0 penetrance in females, even though it's not an X-linked gene. It's on chromosome 17. So, would genetic testing have been really helpful in this month if we had it for this mother in terms of family planning? So my point is that we used to do routine karyotyping. We used to do chromosomes as our first-tier genetic test in ASD. That's now been supplanted by doing these chromosomal microarrays, which now the Ontario government, for example, will pay for these clinical microarrays. And clinical practice guidelines have been developed for genetic testing in ASD. There's 3 different clinical practice guidelines, one from the Canadian College of Medical Geneticists, 2 American societies have produced clinical practice guidelines. All 3 recommend that clinical microarrays replace karyotyping as first-tier testing for genetic testing in ASD. There's a problem, though, and the recommendation that microarrays replace karyotyping is based simply on the better yield for detecting abnormalities that you get with clinical microarrays than you do with karyotyping. But there's a whole arena of testing that needs to occur that looks at the clinical utility of genetic testing. Does it in fact do more good than harm? Does it really influence family planning? How do parents feel about the information they're getting that there's a de novo genetic variant or it's inherited? What's the cost? What's the benefit? There's a whole series of questions for which there's absolutely no data whatsoever in the field of clinical microarray testing for kids with developmental disability and autism spectrum disorder. And there's this really important publication from the Institute of Medicine in 2010 saying that you have to do studies of clinical utility, randomized control trials, to look at the benefits for the true positives and the true negatives and the cost to the false positives and the false negatives. So say we tell somebody, this child does have a copy number variant in this gene and they get it wrong because they're, for one reason or another, what is the cost that's involved? And that whole series of studies has not been done at all in the field of autism spectrum disorder and clinical microarrays. So I just want to emphasize the first paper comes out in 2007, 2011, 2012, clinical microarrays adopted by professional societies as a first-tier test. So a 4-year, 5-year lag between the first publication and translation. But I wonder whether it's premature in the fact that we don't really have studies of clinical utility in order to support first-tier testing in this particular way. Now the translational impact that I think we're all sort of very eagerly anticipating. So one of the important outcomes that we've identified all these different genes that might be involved in the etiology of ASD. And can that lead to new interventions? So Although we've identified the genes, we don't know what the function of those genes are. In order to learn about the function of those genes, we have to do studies with animals, studies, for example, knockout mice. We have to do pluripotential stem cell studies to see how variants, those copy number variants, influence the function of the gene. We have to figure out, okay, how does that affect the growth and the structure of a particular neuron? We need to see whether we can reverse the function of those genes. And I'm just going to give you an example, again, of this Shank family of genes. So here's a paper that was published in Nature in June 2012. You all know that one of the big problems with the drugs that we have for ASD don't actually treat the core symptoms of the disorder. They treat aggressive behavior, irritability. They don't actually reverse the social communication impairments that are associated with the disorder. But what we can do is we can take a mouse, and we can disrupt the function of the SHANK2 gene, which is a close relative of the SHANK1 gene that I talked to you about. And we can look at the behavior of those mice, and we see that when we knock out that particular gene, those mice begin to show real abnormalities, atypicalities in their social development, the way they sort of aggregate together. So in this mouse model, they showed reduced social interaction and communication. They weren't squeaking as much to each other as you would think. The exciting thing is that there are a group of medications, of drugs, that were able to reverse the effect of that genetic mutation, both on the function of the neurons and also the behavior of those particular mice. So those mice that were treated with this mGluR5 receptor medication, their autistic characteristics, if we can use that term to refer to behavior in mice, were reversed. It went away. So this is really the first example of being able to actually develop a medication, or to think about a medication that can tackle the primary symptoms that are associated with the disorder. This paper is a fascinating one, and it's still on. Actually, maybe it's been published, but it's in Nature Online, and it's creating mouse models of tuberous sclerosis and Fragile X. So, tuberous sclerosis and Fragile X, two genetic syndromes, very high frequency of ASD in those genetic syndromes. And these guys from Mark Baer's laboratory created mice that had knockouts in both the tuberous sclerosis gene and in the Fragile X gene. And what they found is that the knockout mutations in those two particular genes caused opposite effects in protein synthesis at the synapse. So if they created the tuberous sclerosis knockout, the mouse looked autistic. If they created the Fragile X knockout, the mouse looked autistic. If they did both in the same mouse, the mouse was normal, because the protein synthesis at the level of the neuron that was caused by the tuberous sclerosis knockout was the exact opposite of the protein synthesis abnormality caused by the Fragile X knockout. So this is the kind of example of what they're saying, is that this is sort of what the protein synthesis looks like. If you've got the tuberous sclerosis knockout, there's reduced protein synthesis. If you've got Fragile X knockout, you've got too much protein synthesis. You put the two of them together, you resemble more and more the wild-type mouse. And again, they had this mGluR5 receptor medication drug that was able to reverse the effect. So again, what's really exciting, seems to me, in this work is that, okay, we're beginning to develop animal models now where it's possible to create autistic-like behavior in the mice and then begin to have medications that can reverse it. And this will, I think, perhaps could provide much more efficacious interventions. Just to draw some general conclusions, is my last slide, is that the discovery of these copy number variants then lead to genetic loci that can be altered by knockouts. And creating knockout mice can help you study the structure and function of neurons and correlate that with behavior. And there's this potential for reversing the losses and gains of function that are created by these knockout mice, and that maybe we can impact the core deficits. So of course, we have to now take this to randomized controlled trials in humans, and that is already beginning. So people are beginning to do randomized controlled trials with these drugs in humans and kids with ASD. So it's going to be really, really interesting to see what happens. So I've been talking about translational medicine. I've been talking about translation specifically in the field of ASD. And one of the sort of themes that strikes me in this whole field is that the process of translation involves a tension between striving for parsimony and appreciating complexity. I think this is sort of maybe most apparent when it comes to thinking about translating all that phenotype research in ASD into the DSM-5 classification system. They've taken a huge body of literature, they've simplified it to come up with DSM-5. That's the process of translation. The clinical microarrays, they simplified it in order to translate it. The problem is, in both cases, perhaps more so with the clinical microarray diagnostic testing, not appreciating the complexity. So there's this tension in translational medicine between thinking parsimoniously and appreciating complexity at the same time. And this, where you are on this tension between these two, I think will have an implication as to where you sort of end up. I think it's a good example of how translation in the ASD field has been so slow for many years, but now with the technological advances, how that pace has become much, much more accelerated. And there are dangers of too much parsimony, for example, dangers of premature translation or delayed translation on the other hand. You're really caught between a rock and a hard place, and I think the next decade is going to be a really interesting time in the ASD field as we try and navigate between these two sets of shoals that I think are going to be very, very difficult. So thank you very much.